Hi, DrBiology here. This is lesson 6 and I'm going to be talking about vaccines, vaccinations, herd immunity and the ethics of using vaccines. So this is within the A-level biology section of 3.2.4, which is cell recognition and immunity. Okay, let's do a quick spec check. So today I am going to be looking at this section here, which is the use of vaccines, the protection for individuals against disease, and also looking at the differences between active and passive immunity. And then I'm going to talk a bit more about the ethical issues associated with the use of vaccines. So before I talk about vaccines, let's talk about types of immunity. So you have both active immunity and passive immunity. So, and these can then be subdivided into natural and artificial. So for active immunity, your natural active immunity is related to immunity after catching diseases. So this is when your B cells produce plasma cells and memory cells. And those memory cells can produce antibodies quickly. You can have active artificial immunity, so that's related to when you have a vaccination which contains a harmless dose of the antigen. Now we look at passive immunity, and your natural passive immunity is when you are in your mother's womb. So you receive antibodies from the mother's placenta, and then when you are born in the first few months you might have breast milk, which also contains antibodies from your mother. Artificially, passive immunity can be when you're actually injected with antibodies from someone else. So particularly, for example, if you're bitten by a snake and you need to be treated very, very quickly to deal with the venom, so you might be injected with antibodies. Now in your exam, they might want you to compare the two types of immunity. So this is a really useful list. So let's go through them. So between active and passive. So first of all, active requires exposure to the antigen, whereas the passive immunity doesn't require exposure to the antigen. Active, it takes a while for protection to develop, whereas for passive immunity, protection is immediate. Active immunity, memory cells are produced, whereas in passive, memory cells aren't produced. And then active protection is long-term because the antibody is produced after activation of memory cells. And then in passive, protection is short-term because the antibodies given are broken down. So here's a little top tip. What I would do is you can learn this list, and basically this list here is the opposite. If you look, it's the opposite of active immunity. So you can learn that and then think about, well, what's the opposite? And then you should have your answer. So as we said, vaccines are an example of active immunity. It's artificial active immunity. So why is it artificial? Well, they contain antigens from the disease by injection or through the mouth, through a pill. Now, the antigens themselves won't cause the disease itself. They're usually weakened pathogens, and they might be just the antigens or parts of the antigens. And the reason that you would do that is that you're stimulating an immune response. So memory cells are produced and they remain in the blood. So if you're infected in future, the result is a rapid production of antibodies. So this can provide protection not only for individuals, but for whole populations. Sometimes you might have booster vaccines as well, given later on, so a few years later, to make sure that more memory cells are produced. So why is it important that if vaccinations are going to work, large proportions of a population who might be exposed to an infection are vaccinated? The reason is that some are just too young, they're maybe sick, they might have conditions that means that they already have weak immune systems or can't have vaccinations, or they might be elderly. And it is therefore important that sufficient numbers of people in a population are vaccinated to protect all in society. As we know as well, more recently with COVID-19, it also reduces the pressure on health services during epidemic and pandemic situations. So vaccine at the same time disrupts the transmission of the pathogen. So that's why at a certain age you would be given, all people of that age would be given a vaccine. This diagram kind of summarizes why it's important for many people to be vaccinated, and thanks to The Guardian for this infographic. So as you can see, if there's a 10% vax rate there, okay, you can see the infected numbers are quite large, and you get a large amount of contact with infected people, so it spreads really easily. So there are a few vaccinated people, and there's a few susceptible people as well, but it's not enough to protect you. And we go through here, okay, and you can even see at about 70%, you're still not fully protected. So vulnerable people would not be protected. So you get to the point where you need about a 90% vaccination rate to ensure that those are susceptible are protected from infection, so the infection cannot get hold of the population. So it's really important that large proportions of a population are vaccinated to provide what we call herd immunity. However, some parts of society have problems with using vaccines, and this could be related to different reasons. So let's have a look at a few key points here. So you could learn these key points, and this is quite a good list of ethical issues. So number one, all vaccines are tested on animals, so that's preclinical trials before human trials. And obviously some people would have a problem with that. They disagree with animal testing. Number two, it's animal-based substances may be used to produce vaccines, so people don't want to put animal-based substances into themselves. Thirdly, testing on humans can cause risk to volunteers with unintended side effects. Four, if the vaccine doesn't work, then some people are put in unacceptable, risky situations. For example, if you were to provide an HIV vaccine, and it doesn't work, and you think it does, then it's going to cause a problem with the spread of HIV. Number five, some people are concerned that vaccines cause unacceptable side effects. So, for example, famously is the MMR debates about autism. I hasten to add there is no scientific link between children having the MMR vaccine and autism, but some people would not agree with that. The other thing is the parents who choose for their children not to have a vaccine, they are still protected by others who have taken the vaccine. And could this be considered unfair in terms of the risk that people are taking to take the vaccine for those who wish not to take the vaccine? And number six, who decides whether someone gets a new vaccine first during an epidemic or a pandemic? So again, with COVID-19, who will decide who gets it first? That is the vaccine. So why don't vaccines always work? Well, pathogens may change their antigens, and that's through a random mutation. They're not choosing to change their antigens. It would be a random mutation. This is called antigenic variability. So before I show you a bit more about antigenic variability, I just want to show you this diagram. So there's two types of antigenic variability. On the left, you've got what we call antigenic drift, where you get a slight small mutation. As you can see, the antigen has slightly changed shape. Therefore, any production of antibodies after a vaccination wouldn't have a complementary fit to that antigen, and therefore no longer deal with the virus. Whereas antigenic shift, which is quite again is quite important in terms of COVID-19. So COVID-19 is a coronavirus. Now there are other coronaviruses out there, but what has happened is that you've found a shift from an animal strain into a new strain, that now affects humans. And that is more dangerous because it's introducing not just small mutations but large mutations, differences, and meaning that humans have not come across COVID-19 before, so therefore it is highly virulent and spreads very easily in a population. So in terms of vaccinations, then this means that the vaccine immediately becomes ineffective as the antibodies no longer recognize the antigen and can't make antigen-antibody complexes. So this means the body has to start from a primary response position again against these new antigens, and therefore a new vaccine needs to be developed. So related to that, each year the influenza vaccine, so vaccine for flu, is actually changed each year, and the most common antigens are chosen to make a vaccine. So they look at the worldwide prevalence of certain antigens in flu, and they decide on which one they're going to produce a vaccine for. So as we know, new strains of virus are regularly formed, so you're having to constantly produce new vaccines. But as I said to you, it's easier if it's antigenic drift than antigenic shift to produce a new vaccine. Okay, hope you found that useful. As ever, please do subscribe if you haven't already to a lot more videos to help you with your courses. Next video is my last immunity video on HIV and AIDS. So hope you found it useful, and I will see you soon.